Okay, well, so thank you very much for the invitation. So today I'll talk to you about some work that we've done looking at the rupture strength of cell sheets. And I'll present to you mostly the experimental side, and Alessandra Bonfanti will talk to you about the modeling right afterwards. So hopefully it'll be fresh in your mind. So I'll take a couple of slides just to remind you of what epithelia do, and then I'll dive into the experiments. So epithelia are sheets of cells that line all of our internal and external organs. They're often just one cell thick, so about 10 microns. And to create a mechanical continuum, the cytoskeleton of one cell is connected to that of its neighbors. And this occurs through a number of specialized intercellular junctions. And today I'll talk to you primarily about the adherence junction, which links the F-actin cytoskeleton of one cell to that of its neighbors, and desmosomes, which link the keratin intermediate filaments across cells. So both of these types of junctions have got roughly the same structure. They consist of a transmembrane protein, which will link one cell to its neighbors. And on the intracellular side, it will link to the cytoskeleton, and often via a number of adapter proteins. So these epithelia are the first line of defense against of our organism against the exterior, and so they'll try and prevent passage of pathogens and waste products, etc., into our tissues. And one challenge to this barrier function is mechanical stress. And that can be mechanical stress that arises through normal organ function. So, for example, in the lung, the alveoli deformed by 20 to 30% at strain rates that can reach 60 to 80% per second when we breathe. In our intestines, when we digest food, the lining of the intestine will be exposed to deformations on the order of hundreds of percent. In our skin, whenever we move our limbs, we create some deformation on the skin, and that can be quite high. So, for example, if I move like this, the skin on my shoulder blades deforms by 60%, and I can do this very quickly. So we easily reach hundreds of percent per second. So it's crucial for our everyday function that this barrier shouldn't break. But actually, in embryonic development, there are some situations where we need these fractures to occur. So, for example, in the Drosophila leg, the Drosophila leg initially consists of a squamous epithelium called the peripodial membrane, which is in red here, which is in apposition with a columnar epithelium that forms the leg. And as this leg extends, the peripodial membrane needs to break. And so this is what it looks like, and this is a movie I've pinched from Magali Suzanne's lab. So this here is the columnar epithelium, and this here is the peripodial membrane around it. And what you'll see is that as the leg extends, this peripodial membrane ruptures, and it then retracts to allow the leg to extend. But overall, I'd say at this stage we know very little about the rupture strength of epithelia and what controls rupture in epithelia. And part of the reason for that is that it's challenging to look at tissue mechanics, because these epithelial tissues tend to be attached to a substrate. And so when you deform the substrate and the epithelium together, most of the stresses tend to be borne by the substrate because it's thicker and it's often also stiffer. So about a decade ago, we decided that the easiest way to try and study this would be to generate epithelia without a substrate. And the way that we do this is that we polymerize a drop of collagen between two test rods. Once this collagen is polymerized, we use this as a substrate to seed cells on top. And when the cells cover the whole of the collagen part of each test rod, we can then enzymatically digest the collagen, and we're left with a freely suspended layer of cells in between two test rods. This layer of cells maintains the normal polarization that you see in epithelia, so that we've got an apical sign which is very distinct from the basal side. So in green, I'm showing a classic apical marker. And this is what it looks like. So we've got one test rod here. Here's the other test rod. The tissue in between is about 1 millimeter in width, and you can just about distinguish some of the cells in there. So we've got tens of thousands of cells, and these are attached to one another only via intercellular junctions. So we then interface this to a mechanical testing system which sits on the microscope stage, and that consists of a manual manipulator here which maintains a reference rod in position, and then we've got a motorized manipulator which allows us to deform the monolayer here, which is within the setup, we've got a force transducer so that we can measure forces at with high accuracy and high bandwidth, and we can image everything that's happening with the microscope on the inverted microscope. And we can image either at the tissue scale or at the cell scale. So to look at rupture, we did some very simple experiments. Essentially, we just stretched the monolayer at a constant rate until it ruptures. And this is what it looks like. The monolayer is here. This is the test rod that's moving. And essentially, you pull, and at some stage, we get a crack that appears, and this crack propagates. And because we're measuring everything, we can actually start getting data which looks like what you would have in mechanical engineering. So we can plot the rupture, the tension as a function of strain. And what you can see is that the tension increases up to a maximum, which is when this first crack appears, and then it decreases as we increase. The first thing you can see right away is that these tissues are very strong, so you can more than triple their length before you get the first crack, and you have a more than quintupling of the length before full rupture of the tissue. So whenever something breaks, you usually want to know where. So if you image at higher magnification, what you can see, so here is one cell, another cell, etc., you can see that essentially you have a rupture that occurs through delamination of cells from one another, and the crack path follows cellular junctions. So in some physiological processes, I told you that tissues need to self-rupture. And one process in which this is particularly important is epithelial to mesenchymal transition. And it's something that occurs normally in development, but also occurs in the later stages of cancer before metastasis. And some work has indicated that EMT may result at least partially from increases in contractility. So what we did is that we took one of our tissues and we treated it with a drug, colliculin here, which increases cell contractility and cell surface. What you can see in this movie is that essentially you have some contraction of the tissue, and then at some stage you start getting some ruptures that occur within the tissue, even though in this case we haven't deformed the tissue at all. So when we measure the tension as a function of time now, what you can see is that this tension increases again up until a maximum, which corresponds to the time when we have these first visible cracks within the tissue. So because this is a very messy drug, we did some experiments to convince ourselves that this was really due just to the action of the motor proteins. So we combined this drug, which is not very specific, with another drug which is much more specific, and this inhibits myosin contractility. And you may not see it, but this movie is actually playing. There's not so much that's happening. But what you can see in this case is that the tension essentially doesn't increase over time, and you also don't get any ruptures within the tissue. But one thing that's different between these experiments and the previous experiments that I showed you is that tension in these experiments is about 10 times lower than in the experiments where I apply a ramp, and the time that it takes for cracks to appear is 10 times longer. So you might even ask whether it's the same phenomenon. And to try and gain some insight into that, what we did is that we carried out experiments at different strain rates, so from 0.1% per second to 3% per second. What we can see in this case is that the rupture tension increases with strain rate, but the rupture strain and the rupture time decrease with strain rate. And that's a little bit surprising, because essentially what it means is that if you go very slow, you can deform the tissue to extremes, but you remain at very low tension. And that was a bit surprising to us, because we'd done some experiments previously to look at the rheology of tissues at these fairly low strain rates. And we'd found that they behaved more or less like an elastic solid. And so in an elastic solid, if you deform more, you should have more tension. But that's the contrary of what's observed here. And in particular, if you go at very, very slow strain rates, like this one that's still moving here, you can reach some real extremes. So this one actually starts rupturing around, I'll try and play it, but you get to more than 10 times its initial length before you see any rupture in the tissue. So it comes around here. So you finally have a rupture once you're at something like 10 times the initial length. So you can take all of this data and you can put that into a rupture tension, rupture time diagram. And what you can see is that you've got an exponential relationship between these two values. And essentially, you've got a trade-off between the tension that you apply and the time that it takes to rupture. So if you've got a high tension, you've got a short lifetime. And if you've got low tension, you've got a longer lifetime. And what you can do is you can now take the rupture tension and the rupture time that we observed in these chemical treatment experiments and put them back into this diagram. And you can see that it falls not so far away from the regression. So that would suggest that rupture in response to active and passive stresses seem to be the same. And this trade-off between rupture tension and rupture time reminded us. Yes, of course.
So, is this suggesting that the passive and active stresses are both acting locally, or?
So I think so they'd be acting on the same structure, presumably, and that is the structure that might be giving rise to.
But it's still not telling you anything about where. Is it global or local?
That's it. It's not telling you about where. It's more likely. So based on other work that we've done, it's more likely to be, so it will act locally, but there isn't a specific region within the monolayer where it I think that's more likely to be a stochastic process. Yeah.
Do you see the rupture at the boundaries when you attach the monolayers often happen?
Yeah, so that often happens, and I think that one reason for that is that you may have noticed that we're not fully horizontal when we apply strain. We've got a little bit of tilt, so your upper boundary tends to be under a little bit more strain than your lower boundary. And also, when we remove the collagen, the cells at the top boundary tend to be a little bit more elongated. So if you remember the video at high magnification, the cells that are close to the free edge tend to be a little bit more elongated than the cells further in the bulk of the tissue.
This question, so do you have movies where it cracks from the center, for example?
Yeah, it does happen. So, for example, the chemical treatment, the first cracks appear within the center of the one. Then in other cases, we also see that.
But on the chemical treatment, like the edge is not, the line is straight, so there's no...
You mean that there's no gradient in tension across the width of the I mean, the free boundaries are a little bit complicated because actually the tissue isn't completely plane because there's a gradient basal and gradient in tension. So there is a curvature, particularly at the edges, it will completely curl on itself. And that curling is dependent on the stress that you put onto it. Yes.
Did you try other caliculing like activate or ROA?
No, we didn't try to activate ROA. I presume it would give you something similar. But yeah, no, we haven't tried. Okay, so essentially this relationship reminded us of what we know about bonds. So if you've got a bond between a ligand and a receptor, if you subject that to high force, it will have a short lifetime, and if you subject it to low force, it will have a longer lifetime. And for collections of bonds that link rigid plates, you have an exponential relationship between the rupture force and the rupture time, which looks very much like what we observe experimentally for monolayers. So this is a simulation, and again, Alessandra will tell you. And the similarity between these two things essentially suggested that the rupture of the tissue at the millimeter scale is controlled by the forces that you apply onto the bonds that link the cells at the molecular scale. So have tissues evolved any way of resisting the deformation? Essentially, if you look at the tension versus strain diagram, you can see right away that there's just change in slope on the order of 50 to 100%. And that's indicative of strain stiffening. So essentially, every extra increment of strain that you apply on the tissue costs you a larger and larger increment in tension. And you can get a better idea of that by plotting the gradient of this curve. And that gives you something that's known as the tangent modulus, which has got a unit of stiffness. And you can see that you transition from a low stiffness at low strain to a higher stiffness at high strain. And this is not insignificant. So if you look, you've got roughly a 6-fold change in the stiffness between the level at 15% strain versus 120% strain. And the other thing that's interesting, and that may relate to some work that Javi might show later, is that this stiffening is actually strain rate dependent. So at the lowest strain rate, so 0.1% per second, you can't see any stiffening happening. And as you increase the strain rate, this stiffening progressively increases. And that's a behavior that I'm told is called shear stiffening. So where does this come from? So usually, my bias coming from what I've done on the cytoskeleton of cells would be to say, well, it's probably the actomycin, but in this case, it isn't. So if you block myosin contractility, if you increase myosin contractility, if you completely get rid of actin, you still get strain stiffening. With temperature, in theory you could, but there's not such a big range that you can apply.
Yeah, but you may expect very big difference between 35 and let's say 41.
41 certainly cells are usually
no,
not
yeah,
so
maybe
happy.
40. I agree, but something like a mild change in temperature, like.
Yeah, is it possible? Yes, but we haven't done. So in any case, in this case, it doesn't seem to be the actin cytoskeleton that's doing something. And the other network that's very important for tissues is the keratin intermediate filament network. And that also forms a supracellular network. So in this image here, I'm showing you the keratins in green, and I've outlined one cell within the tissue. And essentially, these keratins are very dense around the nucleus, and then they radiate out towards the cell periphery, where they connect to other cells. And if you look at this image, it's rather difficult to know where one cell ends and the next cell starts, just from looking at the keratin network. So at junctions, these are connected to desmosomes, and essentially there's several proteins that will link first the keratins to the desmosomal proteins, and one that I'll talk about is desmoplakin, which is the one here. So keratins are interesting in the sense that they're very extensible, so they can extend more than 300% before they rupture. When you put them in networks, those networks display strain stiffening, but most importantly, there are lots of genetic mutations that lead to fragile epithelia. So you have families of people that have got mutations in intermediate filaments or desmosomal proteins, and the type of symptoms that they present is that they'll have cracks in their skin at all of the articulations, which is where strain tends to be high. So what we did is we engineered one of these mutations that comes from epidermolysis bullosa, and that's a point mutation in keratin 14, and you find keratin 14 in some of the layers of the skin. So we overexpressed this in cells, and here I'm showing you a movie of the wild type and the keratin 14 mutants at the same strain rate. And what you can see is you start getting rupture, and here in this case you do have rupture within the bulk of the tissue. And that ruptures a whole lot earlier than this one. So of course you can be more quantitative. And when we look at the rupture tension and the rupture strain, this is lower for the keratin 14 tissues. So we do have more fragile tissues when we disrupt the keratin intermediate filament network. So you can do the same thing, but in this case, rather than disrupting the filaments that make up the cytoskeleton, you can disrupt their interfacing to cellular junctions. And in this case, what we do is we deplete desmoplakin, which is this protein, and this is exactly the same type of experiment. So we've got two tissues that are deforming at similar strain rates. The first crack appears a whole lot earlier in this tissue than and again, you can quantify this by looking at the rupture tension and the rupture strain. We have more fragile tissues. So whether we disrupt the filaments or they're interfacing across cells, we get the same outcome. So just to convince ourselves that the mutations we're introducing have the effect that we think they have, we did some immunostaining. So these are wild-type tissues. This is one cell, and you can see the intermediate filament network essentially radiates out from the center of the cell towards cellular junctions, and you can see it's connected across cells, and you can see desmoplakin at each of the cell junctions. In the cells in which we've introduced this keratin 14 mutation, you can see you've only got very few filaments that reach out all the way to the periphery. Most of it tends to be aggregated around the nucleus or appears in these puncta. So I started all of this discussion by asking about strain stiffening. So we can look at that and compare what happens in tissues that overexpress this mutant versus wild-type tissues. So you can see again here the tangent modulus, and you can see that you no longer have any stiffening in the keratin 14 mutants compared to what happens in the wild-type tissue. And similarly, if you deplete desmoplakin, this is the line in magenta here, you also don't have any strain stiffening compared to the non-silencing control. So essentially, if you disrupt the supracellular network formed by keratins and interfaced by desmosomes, you've got some tissues which are more fragile and that don't strain stiffen. So that was nice, but this is all in vitro using a cell line that's been cultured in the lab for 50 years or so. So you might ask, okay, why is it really relevant to what happens in vivo? So we, with Elias Barriga and Sara Azenia, we carried out some experiments in developing embryos. And the idea here was to select a tissue that is subjected to large deformations. The tissue that we looked at is the lateral epidermis, so it's roughly on the belly of the tadpole, and we're looking at a time when the embryo is elongating in the anterior to posterior axis. So this is the region that we'll be looking at. And so, yeah.
So before you, the mutant of the keratin, you mentioned that is a mutation that you find in patient, but you overexpress the protein, right? Rather than substituting the endogenous one.
Yeah, but it's a dominant. So
it's a dominant negative.
Yeah. So keratins are heteropolymers. So
yeah.
If you overexpress a keratin type, you disrupt the whole network. We'll have the keratins on it. Okay, so what you can see if you look at this tissue is that cells appear to become elongated over time, and here I'm showing you a 10-hour time frame, and you can, the 10-hour time frame, and the type of deformation that they reach is on the order of 40%. So we can look at this at higher magnification. So again here, this is towards the head, this is towards the tail, and you can see the cell boundaries here. Essentially, the tadpoles are expressing a membrane GFP, and what you can see over time is that the cells become elongated in the anterior-posterior, along the anterior-posterior axis. Yeah, this is because it's optical sectioning. So these things here, they're not rather than being a real biological feature. And what our collaborators did is that they then disrupted the network of keratins and desmoplakin by injecting morpholinos into these embryos, so that they now only express very little keratins and very little desmoplakin. And in this case, we saw two different phenotypes. We had a milder phenotype where we see that the cells remain roughly isotropic during anterior to posterior elongation, and we had a much more severe phenotype in which the cells initially extend in the anterior-posterior direction, but then you get a crack that forms within the surface of the embryo. And incidentally, these embryos don't survive much longer. So again here, loss of keratin and desmosomes seems to lead to more fragile tissues. And even though the strain applied is much more modest than the hundreds of percents I was showing you earlier, it's applied over tens of hours. So we have, again, this sort of trade-off between the tension and the time that we subject the tissue to tension. And one idea for how keratins could lead to strain stiffening comes from imaging of the keratins in cells. So if you image them at low strain, or if you image them on glass, you can see that the keratins have got sort of wavy structures as if they weren't subjected to any tension. When you subject the epithelium to strain, you start seeing these very tensed, or these very straight filaments, and that would suggest that they're now bearing load. So at low strain, we would have a low tension because only actomyosin participates in setting the stiffness of the tissue. And as you increase strain, the stiffness will increase because now, in addition to actomyosin, we've also got some keratins that participate. And then at the highest of strains, we've recruited all of the keratins that could participate in the stiffness of the tissue, and we reach a plateau. And essentially what it says is that because of this connection across intercellular junctions, the tissue as a whole behaves a little bit like a fibrous network that stiffens when you apply strain. But this, going back to the strength of the tissues, this strain stiffening leaves us with a little bit of a conundrum. So imagine you've got two tissues. You've got one which is linear elastic, the other one that strain stiffens. And we know everything about the bonds which link the cells to one another. We know the number, it's the same in both. In that case, we should be able to predict a rupture tension. And this rupture tension would predict the strain at which the tissue ruptures in the strain stiffening tissue should be lower than for the linear elastic tissue. But actually in experiments, the rupture strain is larger in the wild-type tissues that strain stiffen than in the linear elastic, than in the keratin 14 tissues which don't strain stiffen. And one explanation for that is that we may need to think about catch bond characteristics for desmosomal coherence. So a catch bond is a type of bond whose lifetime is short. When you apply low force onto it, it reaches an optimum at a given force, and then the lifetime decreases again when you apply more force. So thinking about that, then at low strain, we'd have no force on the desmosomes, so they'd have a short lifetime. Because they've got a short lifetime, they'd make a small contribution to adhesion, and so we'd have a low rupture tension. And if you apply some strain, now you're applying force onto some of the desmosomes, their lifetime increases, they would make a larger contribution to adhesion, and then you'd have an increased rupture tension. So application of strain would do two things. It would stiffen the material, but it would also add effective adhesion between cells. And in that case, because we're adding effective adhesion, then we'd have to recalculate our rupture tension, and that might now explain why we've got a higher rupture strain. So in support of this, if we look at the amount of transcripts that there are for ecoherin and for desmosomal coherens, they're comparable. So there might be enough desmosomal coherens to do what I said might be happening. And desmosomal coherens have similar force lifetime behaviors as ecoherens. The data for desmosomal coherence isn't as complete as for ecoherence, but the ranges look about right. And Alessandra will hopefully tell you a little bit more about this later on. So in conclusion, it looks to us as if these epithelia are tissues which are optimized to resist deformation at high strain rates, but be very deformable at low strain rates. Actomycin dominates mechanics at low strain, but intermediate filaments dominate at high strain, and they underlie strain stiffening. And deformation gives both stiffening and an increase in intracellular adhesion. So there's lots of open questions, like, for example, what underlies the dependency on the loading history, and also what portion of load each of the cytoskeletal networks bears when you reach strains where the intermediate filaments become loaded. So before I finish, I'd like to thank the people who actually did the work. So Julia Duque here did most of the experiments with some help from Annalise Sitcha and Jonathan Fouchard. The modeling we did in collaboration with Alexandre Cabla, and in particular, Alessandra did all of the implementations and all of the thinking about the modeling, and she'll talk to you about that. And Elias Barriga and Sara Azenia did the experiments on xenopus. And I'd like to thank you for your attention.